Good morning, everyone. I'm Giacomo Corleone, postdoc fellow at Regina Elena National Cancer Institute in Rome. Thank you, the organizers, for giving me the chance to share with you the new data produced in the lab regarding the role of the accessibility landscape in B-cell acute lymphoblastic leukemia, an analysis which revealed principle of cancer evolution. My research aims to identify what are called the patient-specific transcriptional modulators, which consider the driver of the cancer evolution. They can be mutations, they can be transcription factors, they can be events which drive the phenotype, and ultimately can be used as a target for precision medicine. So the main idea is to develop targets, drugs which target the patient-specific transcriptional modulators. This because cancer is a very heterogeneous disease which evolve with a combination of evolutive trends spanning from neutral evolution, Darwinian evolution, or Lamarckian evolution. And data from bulk tumor sequencing suggests a genetic evolution. Single-cell sequencing suggests metabolic or transcriptional adaptation to the environment. And for instance, one of the main variables in cancer it's the treatment. Treatment actually induces massive selective pressure on cancer cell population, and subclonal niches can go into a silent state, then adapt and relapse with a new aggressive cancer. Cancer heterogeneity is caused by a genetic component, but mutations do not necessarily explain cell phenotypes. As we can see here on the left figure, the breast cancer case, we have two different diseases, luminal breast cancer and triple-negative breast cancer, but the mutations at driver genes are pretty the same. So same genes that can be affected and driving the cancer. The same for multiple myeloma, very heterogeneous disease, but mutations in different patients are pretty much the same. And RAS, KRAS, these three. And they occur in a patient-specific manner, leading to a similar phenotype. And cancer is heterogeneous because of also an epigenetic component. It's clear that epigenetic regulatory shifts are common in cancer. So the phenotypic divergence is achieved through genetic and epigenetic alterations. And that's the case for B-cell acute lymphoblastic leukemia in children, which is the second hematologic malignancy worldwide, and is prevalent in children, is malignant, and is caused by poorly differentiated lymphoid cells, which produce an aberrant amount of immunoglobulins, leading to bone marrow failure. And the main the classification of this disease is still based only on genetic basis. Treatment for this disease works very well, but unfortunately, 10% of children, in 10% of the cases, evolve with a relapse, with a new aggressive and drug-resistant cancer. So this investigation has three main objectives. The first one is to build a map of cis regulatory activity in BALL. The second one is to estimate the key controllers of the disease progression, the transcriptional modulators, the phenotypical modulators, and then to define the events leading to the relapse of this disease. So we have analyzed 32 32 patients using ATAC sequencing. ATAC sequencing is a high throughput technique which allows the identification of the nucleosome depleted regions, which include promoters, enhancers, and our patients were classified in 4 main categories: healthy tissues, onset, remission, and relapse. Those samples are not matching. And the main hypothesis of this investigation was to track cells at different stages of the disease. So the idea is following the cancer from normal to a new cancer. The investigation lies its foundation on data obtained from this seminal paper from the Greenfeld lab, where it was demonstrated that single-cell ATAC-seq, when aggregated, are very similar to bulk ATAC-seq. So ATAC-seq, every single peak in ATAC-seq can be considered a proxy of cell clonality. It's the amount of cells contributing to that signal, giving the typical shape of a peak. So using this idea, we have developed a computational assay which allows the convolution of the regulatory landscape in a cohort of patients by assigning to each identified regulatory region in ATAC-seq 2 dimensions: penetrance and clonality. As a penetrance, I mean how much a region is active in the patient cohort. Let's see here in the matrix, we have 5 samples. The region is shared across 3. So penetrance, the sharing index is 3, while the ranking index represents the clonality. So how much a signal is clonal in the single tissue. Ranking index in this way. We have a shape. The ranking index span between 1 for the most enriched to 100 for the less enriched. So these 2 indexes assigned to every single regulatory region in a cohort of samples allowed, in this case, the dissection of acute lymphoblastic leukemia regulatory landscape, which provided us a way to identify more than 6,000 regulatory regions, which were modulated during BALL evolution. As we can see here from the figure, it's an unsupervised clustering where we plot the 6,000 identified regulatory regions using normalized enrichment scores, and allowed to identify 4 clusters, and we focus on the main 2, C1 and C2. We can see the population is split between the healthy and the remission, and then the primary tissues and the relapse in 2 different clades. And C1 and C2 was composed by 2,000 regions, which were silent in healthy and remission, and inactive state during the diagnosis and the relapse. So we focused our investigation at those regions and performed a comprehensive data integration with the ENCODE data H3K27 and HK4 monomethylation, allowing for the identification of accessible regions, the enhancers, and specifically enhancers and promoters, and then the dataset from target, which is the most comprehensive data set of transcriptional profiles in acute lymphoblastic leukemia, and the Cancer eRNA Atlas, which allowed to prioritize our target because it includes enhancer expression using eRNA and the relative gene target. So this comprehensive study allowed us to identify three main targets, which were enhancer occurring at MIB, DCTD, and BCL2 enhancers genes. So these three genes are regulated by each one by a single enhancer that evolves during cancer evolution. We focused our attention on the DCTD enhancer because it was interesting. DCTD is a very ubiquitous gene that is expressed in almost every tissue in the body. And from H3K27 acetylation ChIP-seq in our cell line model, primary acute lymphoblastic leukemia, we can see that this enhancer is actively open and regulated, as well as the promoter of the gene. And we know that this enhancer is silent in the totality of the tissues from ENCODE data. So we point our attention on the relationship between the enhancer and the gene expression. And we first identified if the enhancer was interacting with the gene using high-C promoter capture sequencing. We realized that in our primary cell line model, the enhancer here interacts with the promoter of the DCTD gene. So we developed an experimental assay to deplete the enhancer and also the gene, and to see what was happening to the cells. And strikingly, upon DCTD conditional enhancer knockout, we have data that in two acute lymphoblastic leukemia cell line models, that the proliferation of the cells goes down by 20 to 30%. To 30% just by depleting the enhancers. And the expression of the gene upon conditional enhancer knockout was more than 40 to 30% in both cell line. Right now, we are performing several experiments, including the CRISPR knockout of the DCTD enhancer, and also the conditional knockout of the DCTD enhancer in other cell types. So, as a summary of the work we have done, we have mapped the DNA accessibility landscape of B-cell acute lymphoblastic leukemia, and we have provided evidence that the regulatory accessibility landscape dynamically evolved during cancer progression. We have identified a target, the DCTD enhancers, which drive the B-ALL progression, and we have experimental data which provide evidence that DCTD, specifically the DCTD enhancer, can be a target for novel therapy during the relapse. So I would like the team who participated to this investigation, my PI Maurizio Fanciulli, Francesca, who performed all the sequencing, Cristina, who performed the experimental validation, and Stefano, who is a master student in the lab, who performed all the computational analysis, and he will be happy to share. He will present a poster here at the conference, and will be happy to share with you more insights about this investigation. And then finally, our collaboration from the Bambino Gesù Children's Hospital in Rome, Valentina and Matteo, who provided all the sample and the relative clinical annotation. A big thanks to the main funders of this project, the Ministry of Health, the Italian Association for Cancer Research, and the founders of my fellowship, which is the Italian Association for Cancer Research, together with the Marie Curie Actions. Thank you very much.